In 2017, Vincent Racaniello visited Cornell. I made an offhanded comment about the fact that he should make This Week in Virology for immunology. He said, "Sure, you want to do it?" The rest is history. We are now coming up on our 100th episode, and we're going to do something really special to celebrate. A live recording with an audience at the Incubator in New York City on Monday, December 22nd, 2025. The whole crew will be there. Vincent, Steph, Brienne, me, and not one, but two special guests. Gretchen Deal from Memorial Sloan Kettering Cancer Center and Shruti Nayak from the ICANN School of Medicine at Mount Sinai will join us. Just go to Microbe TV and follow the link to the Eventbrite page to get your ticket. There will be light refreshments, good conversation, great science, and a few extra surprises. Don't miss out. Get your tickets today. From Microbe TV, this is Immune, episode number 97, recorded on September 23rd, 2025. I'm Cindy Leifer, and you're listening to the podcast about the body's defenders against disease. Joining me from Cleveland, Ohio, is Steph Langle.
Hi. Hi, Cindy.
Hi.
Great to be here with you.
Yeah, it's just the two of us today.
It's just the two of us.
So we'll power through this.
We will. We miss, of course, our co-hosts, but I think we'll be able to get through this one.
Well, you'll help me because I picked a B-cell paper.
Yes,
and I'm not a B-cell
you
person.
picked a good B-cell paper.
I had to wear my macrophages to help keep myself going today since I'm talking about B-cells and today? I don't know much about them. No. How is it, the weather where you are? Is fall hitting yet?
Oh, yeah. It has been so beautiful the last, I would say, 3 to 4 weeks.
I know.
Really stunningly beautiful. But now it's starting to rain. It's getting cooler, but the leaves are changing. So it's great. I love it.
It's so pretty. It's such a beautiful time of year.
Yeah. And for you all, I mean, it's
gorgeous. The leaves are just almost in full color right now. It's a little
you
early
guys are like
it.
2 weeks ahead of us in terms of when the leaves pop.
Yeah.
Yeah. I'm assuming because it gets a little bit chillier, a little bit faster.
A little bit. But you're also in the middle of the country. It gets a little colder there too.
Yeah.
So.
Yeah.
But so we're missing our wonderful co-host. But in general, if you like what we do, especially today, if just the two of us, if you like what we do, trying to bring you good science from scientists, consider supporting us. So you could just go to microbe.tv/contribute. Any amount is helpful. I know every day I wake up and I'm a little bit more panicked about our world and the information that we're getting, and people really need reliable sources of information. And we really try to be that.
That's why we're here.
Both Steph and I, the other hosts of Immune, as well as all of the hosts of Microbe TV, work really, really hard for nothing to bring you great science. So here we are. And today, lucky for us, we usually do a snippet and a paper. And Steph was ready with a snippet, and I was ready with a paper, even though Brienne and Vincent can't be here. So it all worked out. Yes. So Steph, do you want to tell us a little bit about the paper that you picked?
I do. So I'll first tell you the title. The title is Bacterial Vaginosis Associates with Dysfunctional T Cells and Altered Soluble Immune Factors in the Cervical Vaginal Tract. And the first author is Finn McLean. The last author is Jennifer Lund. I think that's the only senior. Is there, I see a contributed equally. That would be the two last authors. So Jennifer Lund and Jerome Lingapa contributed equally to this work. The authors are from a variety of places. They're part of this KINGA study team that's cited as kind of the consortium that put this cohort together. Fred Hutch, University of Washington, Kenya, and I'm sorry, University of Washington, and also in Kenya, in Nairobi. And so that really leads us into what are they looking at? This is a human cohort study. They are looking at in a population of Kenyan women. And the goal is to understand how the vaginal immunological environment influences the cells and how they respond and what their functions are, particularly as it relates to something called vaginal dysbiosis, or the clinical term is bacterial vaginosis. And in the vaginal canal, it is unlike the gut, where in the gut, you have a very diverse microbiome ecosystem. In fact, the diversity is what makes it considered homeostatic or healthy. But in the vaginal canal, you actually want more like a monoculture. You really want majority lactobacillus, particularly crispatus, as the majority dominant microbe growing and thriving in the vaginal canal. And when you have diversions from that, there are 4 different state types that define what type of vaginal microbiome you have. And whenever you have a vaginal microbiome that becomes more polymicrobial, you have Gardnerella, other types of lactobacillus, one is called Einers. You have these different bacterial state types, and those are more associated with dysbiosis. You have an increase in inflammation, the pH changes. You particularly have that's where you get you have microbes that produce odors that remind you of like that like a fishy smell. That's one clinical implication. And you can do this Nugent score to determine whether somebody has BV. And it's a scoring system that is really the gold standard in clinical settings. It's based on the relative presence of 3 main bacterial types. And that really, again, the lactobacillus, Gardnerella, and Mobilicus. And so determining how much of each you have then determines this Nugent score. The higher Nugent score means you have BV, lower you don't.
So do we know why monoculture is better and how it gets this diverse culture that leads to vaginosis?
Oh, yeah. So one of the reasons that Lactobacillus crispatus, it is able to thrive in this environment because it produces chemicals that help keep a very low pH. And the pH really is one of the main determining factors. Once that pH starts to goes away from not the pH we're used to working with, like 7, it would be something lower than that. And so that bacteria really thrive in that environment. When you get these other polymicrobes that come in, they start to produce inflammatory compounds and the pH changes, and you have kind of these growth of these things that are really deemed to be more pathogenic and creating this dysbiosis, itching, swelling, all of the things I mentioned. And it's a really large problem, actually. 1 in 4 women, I think it's 1 in 4. Is it 3 in 4? But it's a particularly large population of people that get BV. At some point in their life? Correct.
Okay.
Correct.
Okay.
So, and it's treatable by antibiotics, but of the women who get treated with antibiotics, you have a recurrence rate of 1 in 2. So 50% of people can have reoccurring BV, and it's linked to increase in STIs, which we'll talk about today, preterm birth, preeclampsia. And so trying to understand the whys in the hows is a motivation for trying to solve those many issues.
And these other bacteria are considered normal microflora. They're just in the wrong place, or are these introduced from somewhere?
They can, while they could be introduced, it could be hygiene related. They could be introduced because of other dysbiosis happening. If you are infected with an STI, that could create kind of a change in microbiome that allows for some of these other, like the Gardnerellas, to proliferate. So really they can exist there. And this is interesting. You can have a polymicrobial microbiome of the vaginal canal, but not have BV. So that is also true. It doesn't mean that in all cases, if you have polymicrobial, you have BV. But the opposite is usually never true. If you have Lactobacillus crispatus dominant, it is very rare that you would have BV. So some of these correlations don't always hold, but yeah.
Cool.
And what this paper was really interested in understanding is if you have differences in the immune cells and other soluble immune factors in the cervical vaginal tract that would potentially contribute to the increase in STI acquisition that you see with individuals who have BV more likely. And their focus is HIV. Or at least that's what they kind of framed the paper as in regards to some of their findings. So what they did was they had this cohort of 204 Kenyan women. They had many different samples. They had samples from the vaginal tract. They had ectocervical biopsies, and the ectocervix is the portion that is connected to the vaginal tissue. There's the endocervix, which is just beyond the ectocervix, which then would lead upwards. You're moving north. So the ectocervix is in really close contact with the vaginal tissue. And they had PBMCs, they had cervical vaginal fluid and serum. And they did flow cytometry, immunofluorescent imaging, and then they did a Luminex cytokine, chemokine profiling of these different samples to try to understand if women with BV have changes to, particularly as the title suggests, their T cells, as that could have implication for HIV infection. Why would it have implications for HIV infection? Well, HIV infects CD4 T cells, and they use two co-receptors, CCR5 and CXCR4. So those are the main receptors that are also used for viral entry, and the expression of those co-receptors could implicate how susceptible a CD4 T cell is to HIV. So when they went through and compared all of these immunological parameters by their various methods of assessment, BV versus not having BV, they found that they did not see a difference in the numbers of these CD4, CCR5 positive. So again, this is the HIV receptor and co-receptor positive T cells in the cervix or the vaginal tissue. But they found they had the women with BV had higher frequencies of CCR5 and HLA, which would make them potentially more susceptible to a virus that binds said receptor. They also had—
Were you surprised?
Yes.
Like, so if you have vaginal bacterial vaginosis, I would think that you would be having an immune response in the vagina. So would you expect there to be a change in T cells number?
Well, so what they find is that the implication is that the increase in these kind of soluble inflammatory factors, which is well documented, suppresses T cell function, that maybe makes them have an exhausted phenotype. So I guess to your question, would you have expected more numbers of T cells because they need to treat this as like it's a pathogen? It could be that the inflammation is causing downregulation of their function. So maybe they are unable to proliferate. Maybe that's one of the problems with this kind of chronic inflammatory environment. And that is what they find when they kind of dive a little deeper into the types of, so they go into what is different of the CD4 T cells other than CCR5. They found two markers that were increased on these T cells. That one was CD39 and CD101, both of which are markers of metabolic stress, dysfunction, inhibition, exhaustion, and they found an increase in both the cervix and the vaginal tissue of one or both of those types of T cells. And they also found a reduction in what they label as TCF1 positive CD4 T cells, which this is like a marker of stemness. So essentially there were fewer of those, and these would be progenitor cells to differentiate into T cells. To your point, Cindy, that why aren't there more T cells acting? Well, maybe they don't have enough progenitor cells to differentiate. And that's kind of the CD4 T cell bucket. So markers are the marker for HIV and viral entries increased, and then markers of dysfunction, inhibition, exhaustion are increased. They move on to the CD8s, which really should be there to kill infected cells or kill cells that are otherwise dysfunctional. And they showed again an increase in the CD39 CD8 cell phenotype, where they had reduced granzyme B, which is a molecule, a chemical, Cindy, right? It's a chemical that's secreted out. Or is it a protein? It's a protein. Yeah. To help in killing infected cells. So they had lower cytotoxic capacity, kind of painting the suppression-based phenotype. They move on to TH17 cells, which these cells are really known to kind of stick around in mucosal tissues and under normal circumstances help to maintain mucosal barrier function. But when they are dysregulated, they can actually promote inflammation and proliferate and become kind of toxic to that mucosal environment. They, at the same time, found an increase in these soluble immune factors, all the pro-inflammatory cytokines in the fluid, IL-1, IL-17, interferon gamma, and reduced chemokines that would be important for recruitment. So difficult to get them there if they don't have the cues to come there. And that would be things like CXCL9, CXCL10, CCL5. So the local immune dysregulation, it's not just systemic in blood. This is happening locally. And that can be difficult because a lot of these cells, they stay there in the cervix. They're meant to patrol the local space. And if you're not getting kind of new proliferation, as I mentioned, there's a decrease in kind of these progenitor cells, you're kind of stuck in like this pro-inflammatory suppressive environment that would make you susceptible to HIV in this case.
Now, HIV can also go into macrophages, right?
Yeah, there's other, even though it's at a lower percentage in terms of like most likely to infect, they can infect those cells. And they didn't pull out macrophages in this paper or look at that. Because I think if they did maybe like a single cell RNA-seq, they could have done that, but they really use flow cytometry as their
I guess
main
they're just so
way.
vanishingly low in number, maybe. They're there because they're making those inflammatory cytokines that you said they picked up. And
to be honest,
fluids.
I don't know if macrophages increase or decrease with BV. That would be interesting because I would assume they would increase if they're,
I would think.
because otherwise who's producing all these inflammatory cytokines? This just caught my eye as I work with a group here at Case where they focus on female reproductive tract immunology and STI acquisition and have been thinking about this. And so wanted to bring that to you. And
so the bottom line then is these women can get this dysbiosis and that somehow is altering the T cells. And perhaps their argument is during this process, it's upregulating the receptor that would allow HIV to infect their T cells more easily. Correct.
And that they would not have the other cells that would normally kill an infected cell because they are suppressed. So it's kind of, creating this environment where there's more susceptible cells, less cells to kill those infected cells. Problematic.
I would assume that this is not regularly treated in the African population.
I would have to defer to those working at those sites to know. I will say because it is easily treated by taking an antibiotic, I do think they would, depending on socioeconomic access, they could get that. But even in the US, you have 1 in 50% of women have reoccurring BV. So, and there's been clinical trials where you give an antibiotic and you give Lactobacillus crispatus as a probiotic. You're trying to, and then they pre-treat women, they give an antibiotic to clear, the potentially polymicrobial state. Then they give supplanting it in the vaginal canal to give Lactobacillus crispatus. But there's something in some women that they do better with that probiotic than not, but still some women relapse. And why that is, we're looking at it from IgA antibodies that could that play a role? So considering the broad ranging impacts of vaginal dysbiosis on a whole host of women's health issues, this is like one of the key things to try to understand.
That's really fascinating.
Yeah. Huh. Okay. Yeah. So I think when I was looking at those, there's the one more point they made. I guess one thing that was interesting was some of these markers that, CD101 and CD39.
Yeah, they're not things that I know a whole lot about.
That was kind of surprised by that.
So CD39. Oh, I know CD39. So that it's called an ectoenzyme,
Right,
and it's involved in producing
ATP,
adenosine.
Yeah,
which is involved in regulating inflammation.
Right.
So that's interesting. What was the other one? It's like CD39 and CD75 work together.
And then CD101.
CD101
was the other one. Let's see. And that one.
This says surface glycoprotein plays a role in modulating immune responses. So again, kind of regulating. That's interesting because both of them are up or down.
Both of them were up.
And they're both immunoregulating. So it suggests that there is dysregulation of the immune response and they're trying to shut it down.
Which kind of makes sense because I think it has to do with, if you have these bugs that are changing the pH and making whatever cells produce inflammatory cytokines, they're like, the T cells are probably like, there's too much inflammation. We need to bring this down. So then they themselves are trying to shut down kind of this inflammatory setting. So it does make sense that T cells are probably doing what they should, but it's this kind of chronic activation phenotype.
Yeah.
And an inability to turn that off. Yeah. Wow.
I like that.
Yes. Okay. So we're going so that was a little T cell snippet.
Yeah. A little T cells, now a little B cells. So I'm hoping you can help me a little bit. We're going to do a little bit of reminder of Immunology 101 for this paper because I think it's really important. So I would start off by saying, so B cells are, I'll say they're really interesting. I know they're your thing. They are kind of cool. They have a couple really interesting features.
They're just, undergoing Darwinian evolution all the time in our body.
That is amazing.
Pretty amazing.
So the main point of this paper, and so let me pull up the paper. The title of the paper that we're going to talk about next is Somatic Hypermutation Generates Antibody Specificities Beyond the Primary Repertoire. So there's a few authors here. The first author is Teng Zhu, and the last author is Dwayne Westman. They're
at Harvard.
Yeah, but I don't think, oh wait, there is a contributed equally. Okay, so I don't want to leave out the first author.
Yes.
Is Avanish Gautam? Gautam?
Oh yeah, because number 7 is contributed.
Yeah.
Equally. I see. Yep.
Okay. So, and this was published in Immunity recently over the summer. And I saw this paper and I was like, oh, what does that mean? And I just glanced, I was just reading abstracts of things and I read the abstract and I said, wait a minute. My brain went, do not understand.
It very much went like, this is not what I learned from the textbook.
Yes. It's like, I know it's been a long time since I took my basic immunology class, a little too long to want to admit how long that is. But I said, I thought that things worked this way. So B cells during development will rearrange their B cell receptor genes. And so remember, B cell receptor is on the surface of B cells. And then those B cells, when they get activated, they will secrete that antibody, that same specificity. Okay, so they do their VDJ recombination during development, and that creates a repertoire of B cells, each that have their own B cell receptor that has been modified, and each has its own specificity.
Right. And this is happening before it even sees an antigen.
Before there's any antigen.
Right. This is RAG, the enzyme.
RAG dependent.
Bone marrow. Right.
Yep. Okay. So you rearrange your heavy chain, you rearrange your light chain, you pair them together, you get signaling and the B cell survives. And that creates like an incredible number of different specificities. You can correct me, but I looked up numbers roughly in the range of 3 times 10 to the 11th. So we're talking hundreds of billions of different specificities. Yeah,
so you're just ready to recognize anything that the world could throw at you.
That's what I thought.
Potentially.
Right? I mean, it's not infinite, but it's pretty big deal, right? Then when something infects, any antigen comes in, there's a couple of things that have to happen. You have to activate a T cell. So a T cell has to get a T cell receptor signal. They could get that through antigen presentation by an APC. That could be a dendritic cell that will activate, but it could also be a B cell. Because B cells can be antigen presenting cells.
Right.
But my understanding was B cell antigen antigen presenting cells present the antigen that their antibody detects.
Correct. Yes, that would be.
That was my understanding of how this works. So B cells are not phagocytic cells. They don't just eat everything around them. I'm sure they're doing pinocytosis and a little, receptor mediated endocytosis. Sure. But the primary mechanism that the B cells take up antigen is through their B cell receptor. And the reason why is because you don't want to make antibodies to things that you are not getting a signal for to tell you that that's what you should make antibodies to. So basically, the B cell, a checkpoint, I understood important checkpoint to be the antibody on the surface, the BCR, binds to the antigen, brings it in, digests it up, and presents it to the T cell. And then the T cell gives it back the help.
Right.
And that's how we have this check and balance, because B cells don't want to just willy-nilly be presenting lots of different antigens.
But also, just to bring in the context of checks, there are other checks.
Yes.
Even if the B cell gets past this T cell, you need T cell help and stimulatory. You have ways in the periphery to deal with those cells. But you're correct. This was thought to be one of the main checkpoints.
Yes. And then B cells can do really cool things once they get those signals. They can switch the type of antibody they make. So you mentioned IgAs before in mucosa. Those can be really important because different antibodies have different functions. But the other pretty amazing thing is that B cells are really the only cell in the entire body that once you have a set genome, they can actually muck with it.
Right.
And so only, quote unquote, at the B cell locus, although we've done a paper before where we found they can actually make mutations in other parts of the genome. It's just much more frequently happening at the antibody variable region, but they start messing with the sequence at the variable region. Yeah. So it's called somatic hypermutation.
Yes. And it's this amazing enzyme, activation-induced cytidine deaminase. What does it do? It deaminates cytidines.
Yep.
Yep.
And that leads to
somatic hypermutation.
Somatic hypermutation. So basically you have a sequence of an antibody and it changes the sequence of the antibody. Why would you want to do that? My understanding in the textbook definition is because you want to refine the specificity. So something that it already binds to, you can make it bind better. So kind of like putty, where you kind of fit even better to whatever groove you have there. That was my understanding. Now, this paper kind of blows that whole thing out of the water by saying that another reason why you might make these is to create entirely new specificities that didn't exist when you did VDJ recombination and had your original repertoire.
I know I have to, it's suggesting that there may be a reason you want to diversify things even more after the cells exit the bone marrow, after RAG has already acted upon to join the VDJ segments before the antigen even comes in the picture.
Or as an antigen comes in the picture.
As the antigen comes into the picture.
Yeah. And
right, because it's in response to, in their models, in response to the antigen that they're administering. Yes.
Yeah.
Right.
It's still in response to vaccination. So it's in the presence of an antigen that you're then going to generate these new specificities. And there was actually some inconvenient pieces of information that were already out there about this. So you can have development of antibodies that detect tumors, for example, when there was no detectable recognition of or interaction with any tumor in the animal before.
I'm so glad you brought that up, because tumors is what I was thinking. I mean, I know we talk about can we evolve mechanisms to deal with tumors because it happens when you're older. It's not like a viral infection, but that would make sense as a way to deal with a tumor where you don't have you don't have a antigen, but you still need a way to diversify a new gene sequence, right? To something totally unseen before.
Right.
Right. Not antigen specific.
Right.
Right.
So the other weird thing was that people have done things where they have antibodies that have developed to viruses and they look at the mutations that were made to the antibody complementarity determining region, or the specific region that binds to the antigen.
Right.
And they try to mutate it back to the germline configuration, or whatever not before somatic hypermutation. What was the sequence? And the prediction should be that you could still bind whatever virus it is, but you just bind it less well.
Right.
Turns out in some of these instances, there's a zero binding to the virus.
Right. Right.
So how does that happen? Like you now have an antibody that binds really well to a virus, but it was never able to bind the virus before it went through somatic hypermutation. So again, that suggests this really odd thing that doesn't fit with the textbook.
Right.
That apparently B cells somehow are able to make brand new specificities that never existed in their repertoire that they developed during development.
Right. That you have, they call these bystander B cells. They're non-specific B cells that are mutating in response to an antigen it should not care about.
Right.
Right.
And one would think, well, we don't want to do that, right? You don't want to just willy-nilly start messing around with B cell receptor sequences for things that are irrelevant for whatever infection or immunization that you have. But we do have selection mechanisms, right? So if
you still need secondary signals, it's
right.
Yeah. Maybe it's not to say that if you were to create this, allowing for exploration of many mutational options, that necessarily is going to lead to autoimmunity. In fact, if this is true, it clearly doesn't. And so it was able to continue as a function.
Yeah.
The other thing I want to mention about the viral piece of information, I think a lot of people kind of excuse that away based on sensitivity of binding assays.
Yes.
They said, well, just because your assay can't detect it doesn't mean it doesn't bind. They call it like dark antigens.
Yes, exactly.
I thought that was kind of neat.
So dark antigens or cryptic is one explanation for how this could happen, right? Mm-hmm. And the other explanation for how this could happen is what these authors call affinity birth.
Affinity birth, yeah. Mm-hmm.
So it's the birth of affinities that didn't exist for an antigen in the original repertoire. Okay, so the big problem is, though, how could you ever address this kind of question? Like this is not an easy type of thing to address.
Right.
What you want to know is there a way to make an antibody to one thing?
Yep.
And then immunize with something else and see if you can create new antibodies that can recognize the second thing.
But it has to be the case that every sequence of every BCR has to be specific to one thing. Right. It has to be the same sequence.
Yep.
No competition.
No competition. Because you have to be able to detect that new affinity that arises.
Right. Because you're always comparing when you're sequencing back to this original sequence.
Yep.
Right. Okay.
So lucky for this group, they have this. So there are mice that are made that have B cells that have a clonal B cell receptor, and they do this on a RAG knockout background. You mentioned RAG is important for VDJ recombination, so they can't do VDJ recombination. They can only express, quote unquote, the antibody that is put into their genome to express. So they can make one antibody, one and only one antibody, and it has a particular specificity.
And any somatic hypermutation that is acting upon it is acting upon this one sequence they start with. It's a hemagglutinin. It's an influenza protein that they use. It's acting on that.
Yep.
Only. Yep.
Yep. Okay. So that's great. But they also need some T cells to put in.
Right.
And so they need to mix T cells in. And they actually had a prior paper where they showed this affinity birth, and there was a stink about it. People were like, not true. You're using clonal T cells that see like OVA antigen and clonal B cells that see OVA antigen. You put it and you forced it in the system. And of course, something's weird is going to happen.
It's too contrived.
Anyway, I think it makes HA antibody and OVA T cells, whatever. So they said, okay, no, we're going to use polyclonal T cells. So the T cells are unrestricted in their development. They have all the different specificities. So presumably the specificity for any one thing is exceedingly low because that's the way this works, right? And then they have this monoclonal B cell population, but they need a way to be able to look at the cells and look at the antibodies. And the cool thing about this is they have a couple of really neat tools. So I kind of looked at this the way, like, so let's take you, Steph, and we're going to put a clone of you on this side that has blue shoes on and a clone of you on this side that has red shoes on. And you can see the difference because you can look for the shoes. So these are called chongenic markers, and they have the mice. The mice have chongenic markers. So you can see whether those clonal B cells came from one, your donor or a different thing. And you can start to mix things together. So you could take a mixture of normal wild type B cells in one clonotype, and then you could take the chongenic marker. And then you could take the clonal B cells that have another chongenic marker. You can mix them together, put them into a third mouse, and then you can kind of pull them back out and see who's who.
Right.
Do they have germinal center phenotypes? Do they look activated?
Right.
Are they making antibodies? But I thought the coolest part of this was they also were able to differentiate the antibodies. And so I kind of look at this, think about this, where like if you're handing out flyers and you hand out a white flyer or a yellow flyer, right? And only one clone can hand out white flyers and the other clone can hand out yellow flyers. Now you can kind of figure out who it came from.
Right. Right.
So you can not only track the cell and the activation markers on the cell, but you can track the antibodies that those cells made. Right. So you can say your blue shoe wearing Steph only handed out yellow flyers. So you can track the flyer back to the person. So you can track the cell and the antibody. And so this system was, I think, incredibly powerful and allowed them to do the experiments that they did that really nailed this down. And I thought it was pretty convincing. I don't know how you thought about it.
I think we can talk after about some of the potential critiques, but let's go through it because I think it's really, it's very clever. I really liked how they approached it.
So how do they do this? They did what are called bone marrow chimeric mice. Mm-hmm. So they took those HA or monoclonal B cells that made antibody only to HA, and they also did a whole bunch of other experiments I'm not going to go into. Make sure it only bound HA. It did not bind OVA. It did not bind pigeon cytochrome C. It did not bind chicken gamma globulin, which were the other things that they would immunize with and see if it can make. So this antibody really bound HA and nothing but HA. Didn't have any reactivity with any of the other ones. So it's very easy to see now if you immunize a mouse with something different like ovalbumin, if all of a sudden you have antibodies to ovalbumin, they could only have come from the HA antibodies that you put in.
Naive B cells. Because
that's the only way they could have come.
They're not these B cells that exist in the RAG1 knockout mouse are all naive. They're not memory. It's difficult because if you hear they're HA specific, you think, because they're antigen experienced, so they're memory. But no, they're HA specific because they put that sequence in them. They did not choose that, and they're not being acted upon that in these immunization strategies where they're giving OVA, APC, CGG.
So they're going to mix those clonal B cells that recognize HA with wild type B cells. And why would you do that? Well, you need to be able to see whether there's something wonky with the system. You want to make sure that if you immunize with OVA, that wild type B cells in that environment can still make OVA specific a reaction.
Because if they don't, then you would say, well, there's something funky with your
So
mouse.
The prediction based on the textbook would be if you take these cells, the HA specific B cells won't do anything. They'll just hang out and their antigen's not there. So they keep moving around the body looking for their antigen their whole mouth life. That's what happens, right?
And maybe one could say, well, what about like cross-reactive epitopes? Which I think is a fair comment. They kind of tried to screen that out with this binding where they said, well, HA doesn't bind these, but, so that's how they kind of try to screen that out.
One could always argue, is there a teeny tiny little bit of reactivity? And that's
I don't
think you can ever rule that out.
Point with it, and they do address. So I think one of the critiques is like, well, is this just specific? I don't mean to be jumping ahead of you, Cindy, but we'll address it. But is this just specific to the sequence of this HA?
Yes.
Fab.
Okay. So the spoiler alert is no. They use two different model systems. Right. I'm not going to go into the depth of each model system.
Sure,
Mostly I wanted to set this up and kind of set the scenario and tell you a little bit of the data that they did. I'm not going to go through every single individual figure. But what they did do, though, is they took those monoclonal B cells, they mixed them with some normal wild type B cells, they parked them in a mouse so they could find the original clonal cells, they could find the antibodies, whatever. Then they start immunizing. They don't immunize with HA. So if you immunize with HA, presumably you're going to get lots of HA antibodies. You have lots of B cells that are specific for HA. But they immunize with OVA. And the prediction in the textbook would be that you get a little bit of response because you have wild type cells there. And there should be some OVA specificities in all of that original repertoire that they made. But that the HA cells are not going to see the antigen, they're not going to respond. Right. And they did it different doses, so different amounts,
that was
which is interesting. So if you mix them 1-to-1, there's a lot, there's equal numbers of the clonal ones and a lot of the polyclonal ones. If you start to add 100 times more of the clonal to the polyclonal or 1,000 times more of the clonal to the polyclonal, basically what you're doing is there's vanishingly small numbers of B cells that should be reactive to ovalbumin.
Right.
But lots and lots of B cells that would be reactive to HA or all the other potential things.
Right.
So you've really, really limited that repertoire.
Yeah.
And what they see is that if you look for antibodies, if there's wild type cell, if you look for the antibody, the white flyer that the wild type B cells make, they make antibodies to OVA. If you immunize them with something else called APC, they make antibodies to that. If you immunize them with something called CGG or chicken gamma globulin, they make antibodies to that. But in the same exact mouse, you can look for the antibodies made by the clonal cells because they're the yellow flyers. And OVA, nothing. APC, nothing. And that's expected. But interestingly, with CGG, they see a few mice that make antibodies that react with that antigen. Now, that is not the right B cell. It's not the wild type B cell. It has to be coming from the B cell that is monoclonal for HA that now makes some sort of antibody specificity that now recognizes CGG.
Right.
There could be a lot of things going on, but what the first thing they do is they say, okay, well, what about those B cells? Did they actually go into a germinal center?
Right.
Did they take on a phenotype of an activated germinal center B cell?
Right.
Did they mutate their sequence? So do they have evidence of somatic hypermutation? And do they make antibodies that have high specificity? And the answer to all of those is yes. So they can see increased numbers of cells that have a germinal center phenotype. And you know the markers better than I do there. But let's just say they have the phenotype.
GL7, CD38.
Yes.
Yeah.
And then and they can track. So the wild type cells obviously do. Because you would expect them to, but they can look specifically at the clonal B cells that are specific for HA, and those also have the germinal center phenotype. Yeah. And then when they look and they do sequencing of the B cell receptor in those clonal cells, which they can sort out and look specifically at those, and they can look in the different conditions where they've immunized with OVA or APC or CGG, and they see mutations.
Yes.
So these cells have gone into germinal centers and they have gone through somatic hypermutation, and the mutations are not minimal. They're like up to 20, 25 mutations.
I mean, they're moderate amount of mutations. It's more than I would have expected for a cell that should not be able to recognize that antigen.
Right.
And Cindy, when you mentioned that they saw a couple mice with the CGG immunizations that they saw antibody titers, but not in the OVA or APC. However, they did see antigen-specific cells. This speaks to this kind of the distinction between you might not make detectable amounts of antibody in the blood, but yet the cells are still going into the germinal center and being mutated upon. Is that fair?
Yes.
Right. There are more individual animals that have antigen positive cells than they do seropositivity.
They all seem to have, right?
Right.
Yeah.
Some more than others.
Mm-hmm. They do go in the discussion and say that the CGG, I think, was the largest of the antigens. And they said that the ova is the smallest. And so there may be something about size of the antigen that may influence whether or not it's more likely to get taken up by the B cell and induce this non-antigen specific response.
Okay. Yep. On the same page as you.
Okay.
Right.
So one could argue, well, there's these polyclonal B cell population. Maybe there's stuff that they secrete that they cause this reaction in the non-specific B cells. And it's just kind of like a bystander weird effect. So they said, okay, well, what we're going to do is we're going to take out all the other B cells. So they take, but you still need the T cells and you need the other parts of the bone marrow. So they take quote unquote wild type bone marrow, but from a mouse that's called mu-MT, so they make no B cells.
Right.
So the mutation, there's caveats to that because they say they make no B cells, but I read they do actually have some B1 B cells and they have some levels of naive IgMs because it's a or not IgMs, but they can skip the IgM and make some other antibodies because it's a mutation in the IgM, the heavy chain locus.
Okay.
So there's some weird
I know
things
that
I read can happen.
And in figure, we'll get to this, but figure 3 with the B18 3-83, they say that almost all B cells express this, but not all. So there is some like maybe leaky phenotypes, but I do appreciate that they're consistently reporting this. And it's not like a small effect. I mean, they're seeing numbers of mutations that it just like a leaky couple B cells wouldn't maybe explain. That's kind of how I understood it.
Yeah.
Yeah.
So they repeat their whole experiment. They put in the HA clonal B cells mixed with these mu-MT bone marrow, and then they immunize the mice. And the prediction is like if you take out any of the other competition, maybe it has more of an opportunity to make these new specificities. And there's caveats there because you are artificially making the system. There's only B cells now, only quote unquote, air quotes, only B cells that can develop any specificity for the antigen have to develop from this monoclonal HA antibody.
Right.
So it's again, going back to a bit of artificial system. However, in this case, they all made detectable serum responses.
There was more than their first experiment.
Yeah. So they were able to get OVA-specific responses, PC-specific responses, and CGG-specific responses detectable with the serum antibodies. So those HA antibodies were able to mutate and now gain specificity for these antigens.
Right.
And then they did a whole lot of stuff that I don't necessarily follow, and you as a B-cell biologist probably get it a little bit more, but they made these like phylogenetic trees. So basically they single cell sequenced the BCRs of these and they looked at how they developed.
Right.
And so they developed basically multiple different times. So it wasn't just one linear path that this mutation then led to that mutation, and then it developed a specificity for OVA. But they were able to mutate in different ways to get the specificity to OVA or to PCC or to CGG. And they also looked at like the mutations that were replacing versus non-replacing, so synonymous and non-synonymous mutations, and there was a preference to the ones that changed. And they also cloned the antibodies that these B cells made. So these are HA clonal specific B cells that they now, naive B cells, they put in, they immunize to something else. Then they pull them back out and they collect the antibodies, clone the antibody sequence, made the antibody, and showed that it definitely, absolutely had specificity, reasonably high there, for that new antigen. So the HA antibody now binds OVA with a specificity in the nanomolar range.
Yeah. And so it's helpful to do these type of recombinant monoclonal experiments because you can always, I guess, when you're pulling out serum antibodies, what B cells did they come from? You don't know. Now you know what B cells they came from. You know the sequence, and they made a bunch of monoclonals. I don't think 100% of them bound, but a lot of them did.
It was close.
It was close.
Yeah.
Yes. And so what I understand, the data where they found there was no clear path, that the mutations were happening. There were different kind of pathways, lineages that they were experiencing or being acted upon that suggests that this isn't like an antigen specific driving. When you look at those phylogenetic trees of those BCRs, they're very long because they're not, there's not multiple and they're not short. So I think this is framing this argument that, and we'll talk about it more, but like this is kind of intrinsic acting upon the BCR in a non-antigen specific manner that would have driven it down, just like one linear pathway always. And in fact, they're not seeing that.
So it's not this like idea of refinement, but it's the idea of like, let's mutate the crap out of it and see if anything sticks.
Right.
And it does.
Which this sequence intrinsic bias would drive mutational outcomes and drive more diversity outside of what we would traditionally think of as like antigen specific. SHM. Yeah.
Yep.
Yeah.
And they were able to show also that, they had these clonal antibodies, they had a whole bunch of different ones, and they did cross competition ELISAs. And they could show that there were 3 clear epitopes, at least for OVA, and then 2 for PCC and CGG. It was kind of like all over the place. But they could show that there were like specific immunodominant epitopes that these antibodies were directed against.
Which, so there are, I think what we did know is that somatic hypermutation, it is not evenly acting upon every portion of the sequence. There are hot spots and cold spots depending on where the cytosine is in the sequence. So that wasn't new, but I think just the idea that those hot spots also exist and are acted upon in a non-specific B cell.
Yeah.
Or in a non-specific fashion. Yeah.
So basically at this point, they're just saying like a polyclonal T cell setting. So this is when the T cells are normal. A single antibody sequence can acquire a new ability to bind new antigens, different antigens at multiple epitope sites. So really you can birth these new antibodies from an existing antibody that has completely different specificity.
Right. Right.
Which, I mean, it's pretty wild. So you mentioned earlier the other model system. One could very easily say this is some weirdo thing that happens just with the HA antibody, but they used a different antibody called B1-8/3-83. But those mice, but those mice have also a clonal antibody, and this antibody actually isn't specific for anything.
Yeah,
they said.
Right.
So it's kind of like a isotype control antibody. Basically, it has no specificity for anything they can measure. And again, when they immunize with OVA, APC, or CGG, they are able to develop new specificities. They have detectable serum antibodies. They have germinal center phenotypes. They have evidence of somatic hypermutation. They bind to different epitopes. So it's not just the one model system. They also did some really complex things called a logogram, where they look at what mutation, like what amino acids are substituting or changing. And basically, what I got from this is that there are those hotspots you talked about. So there's CDR3 frameworks. Right. Most of the mutations happen in those frameworks during somatic hypermutation. So it fits. The more competition, the less it happens. So the frequency of mutation is higher when it's like a 1-to-1 ratio or 1 to mu-MT B cells with no BCR. Right. So it definitely makes you think about what would be happening in a normal person under a normal situation where there's a normal repertoire of B cells. How frequently is this going to happen? Probably exceedingly low, but these data suggest that it can happen.
Right. Right.
So the last piece, I'm not going to go through every single piece of data, but the last piece I thought was interesting. And I will say that if you are an aficionado of these antibody and BCR sequences, you should go check this out. I don't know that I fully understood the figure the way they presented it. So this is the one where there's a whole bunch of different dots that are different colors, and there's affinities and whether the mutation is shared amongst the BCRs that were discovered and sequenced when they immunized with different antigens and things. The bottom line that I took away with this was that there are actually shared mutations, and they're more common than unique mutations.
Correct.
In the CDR3.
Right.
So what I took from this is that there are common mutations that kind of maybe cause a degenerate antibody, so a less specific binding that allow it then to go down different paths to try and gain this new specificities. That's sort of how I looked at it. So that they're very common amongst all three. So there were actually mutations that were found in B cells, those clonal HA B cells, after immunization with all three different antigens. So there were B cells that had the same mutations. So there's somehow those mutations are very common, and they allow them to drive down these different paths to develop these new antigen specificities.
Which I mean, to me, this actually helps me, puts validity to this paper, because these hotspots are not, we knew about these hotspots. So it kind of makes sense that the majority of mutations are from hotspots that are just inherently mutable based on their sequence, not from antigen-driven selection. So it fits within the other biology that we already knew about hotspots. It's just happening non-antigen specifically.
Which is still...
Yes.
Yeah, it's still weird, right?
It's so weird. And
so go ahead.
Well, again, we talked about tolerance, because this seems to be happening just, I don't know if a lot is the word I don't know how to describe it, enough that there's still pretty high bystander tolerance. Even non-selected B cells that can accumulate mutations, they're not being eliminated because they can detect them.
Right.
So how does that work?
I don't know.
Maybe they're just not abundant enough. And as we know from this paper, when you remove competition, there's more of them. So the fact that we exist as a polyclonal, we as adults have polyclonal B cells, they probably don't ever become high enough in the percentage of B cells to have to deal with them, to become a tolerance issue, I guess. Maybe. Yeah, but that's how I understood the color coding of that graph as well, Cindy, that the dot size was how frequent the mutation was, the color was how widely it was shared.
Whether it was shared. So basically there's purple ones that are unique. So those would be a unique mutation, and they were very rare.
They were very rare.
So it was much more common to develop. And they came up with this thing, it was a privacy index. And again, if you're a B cell aficionado, you might look into that. I'm not sure I totally followed all of it, but
I think it's saying that antigen specific sequences have enriched privacy because it comes from common mutations rather than requiring rare mutations. Right? Did I say that right?
I think so. I think so.
I know it really is. Every figure of this could be its own. But no, that I mean, it's kind of leading to telling us how this is happening. It's being acted upon in these hotspots. It's sequence intrinsic. It's common. And now they kind of want to learn about T cell co-stimulation.
Yeah. So
how would you think about that?
We know that the B cell somatic hypermutation requires T cell help. So one of the things they wanted to say was, could they manipulate the system a little bit and cause this to happen more? And one way to do that is checkpoint blockade.
Blockade, yeah. When is that kind of? I guess that's not what they did in the past paper. They just did a monoclonal TCR.
They just used a monoclonal TCR.
They didn't remove them like they're doing.
Yeah.
Here.
So in the prior paper, if I understood it correctly, was they had the HA antibody clonal B cells. They're going to immunize the mice with ova, but every T cell sees ova.
Right. Right.
So any B cell that spuriously took up some ova and could present it, that T cell could then potentially give it help.
And in this scenario, when you say CTLA4, the T cells are still there. They just can no longer, they're increasing their help.
They're increasing their help.
They're still
right?
polyclonal.
So they're blocking
still there.
the negative signals that CTLA4 would normally give.
Right.
And they're asking, can that then promote the generation of these new specificities? So if you remember, when they did the first experiment, we explained they took the HA clonal B cells and they mixed them with wild type B cells. And there really was almost no serum reactivity, but there were germinal center B cells. Mm-hmm. This time when they block the inhibitory signals on T cells, now they can start to see some serum reactivity to PCC and a lot more serum reactivity to CGG.
Right.
But still no reactivity of the serum to ova. For the yellow flyers that were coming from the clonal B cells, right?
Yeah.
The wild type B cells do normal stuff. With or without checkpoint blockade. So their argument again is like there's a gradient between ova, PCC, and CGG, where CGG has the highest propensity to develop, the antibodies will develop against that particular antigen. And they have ideas that that's the largest one, and maybe that's why. But even so, even when you can't detect serum antibodies, there's still a lot more germinal center B cells in the ova immunization condition if they block CTLA4.
Right.
So it's still suggesting that, yes, T cells are, one, critically important, and two, if you ramp them up, they can promote this, which got me thinking, like when we're using all this checkpoint blockade treatment, could this be happening more?
Which would be good, because if you're trying to deal with the many foreign antigens that a tumor presents you with, this would be helpful.
Correct.
It would be interesting to understand if one of the ways CTLA4 improves clinical outcomes is not only helping T cells do their thing, but this mechanism as well.
Right.
And they had greater diversity as well.
They did.
Right. Okay. Which is to think about that a little bit more. Fewer shared mutations, more unique ones. Yeah. I'm just trying to understand kind of what that tells us about the mechanism of how this is happening.
Yeah, I don't know.
It's still a little fuzzy, which I'm sure is what they're working on now, right?
Yeah. So what you're saying, what they stated was that CTLA4 blockade led to fewer dominant mutations and more unique mutations. So I guess that it is changing the path to get to the antigen specificity. Because
so then the enzyme's acting upon it more because it's a higher mutational load. So then AID would be more. I don't think they talked about I
don't think they did.
AID freak activation or function itself. No. Okay.
I think when we talked about it before in those phylogenetic trees, remember there were there were a bunch of shared mutations. So there was some common event, some common mutations that would then allow it to go down different paths to create different specificities. And I guess their argument is with the CTLA block, there were fewer of those and more unique mutations. So there were different paths that a B cell somatic hypermutation could take to get to the antigen specificity.
Yeah.
I don't know what that means in the grand scheme of things, really.
I mean, to me, it would help in if you're dealing with a tumor. I think that sounds good, like a good thing. Okay. Interesting. I will say one thing that kept me coming back is, and they did have an explanation, but it wasn't the same for all antigens. So it is antigen specific in a way.
So it's
dependent.
It's antigen characteristic.
Right.
So the characteristics of the antigen influence how much how
well it can be presented by a B cell?
Could be how well it's presented by a B cell. I was just going to say the nature of the antigen would determine how often a birth from an old specificity can get to that antigen.
Yeah.
I don't know. That's kind of a crazy way of saying it.
No, I agree. And it must be, and maybe if we like had lined up the sequences, it also must be like how many, did they talk about like how many hotspots are in each of these? And the fact that CGG is larger means there could just be more hotspots.
Yeah, there's probably more
potentially.
hotspots, yeah.
Okay.
So that's the data that I wanted to present. I think it has a couple of interesting things. I mean, really, we thought before this, at least I certainly thought, that in adults, mammalian AID and somatic hypermutation is there to support refinement of specificities of existing antibodies. And this really is, quote unquote, rewriting the textbook to say that there is this potential to take totally nonspecific antibodies and birth new specificities. So what does that mean in the grand scheme of antibodies? It means that they show a picture where there's this whole, universe of potential antigens that we could come in contact with, and a certain, like, thinking of like all the stars in the sky that we can see in our galaxy, but then there's other galaxies, right? So we can see, we can make the repertoire of 3 times 10 to the 11th different specificities that will see things in our tiny little part of the galaxy. But there's actually lots of different things that we don't and can't possibly make specificities to, because there's just too many. It's like infinite, basically. And what this allows you to do is go outside of what the repertoire encoded to create new specificities. So I envision 2 things. One is that this is important for potential new pathogens that come out that the human race has never seen before. And perhaps we have some antibodies that have specificities for it, but maybe not. There's always a possibility that there's not, right? And this would provide an opportunity to make specificities to practically anything.
Right.
The dark side of that, though, is that I feel like there's also a potential for this to have contribute to develop of autoantibodies. Yeah,
totally. Yeah.
Because if we selected things out, we didn't talk about that part. When you go through your development of VDJ recombination, we get rid of some of those specificities that are recognizing self so that we don't carry them around generally, so that we don't have autoimmunity. There's genetic components to that as well. But then could this be a reason why occasionally you might generate antibodies that can react with self?
I mean, I think it speaks to the evolution of a species would require many ways to deal with things that could kill us, but you can't go too far because you could kill yourself, right?
Yes.
This could have been evolved a way of bystander B cells to create more diversity. But if that were to get out of hand, especially in a case where you don't have, in this model, competition from polyclonal B cells, that could potentially, I would be very interested if they could find a way to force this to become an autoimmune disease. Like, do you see what I'm getting at?
Yeah. So maybe if you did it so your HA monoclonal B cells in, for example, in EAE model, where you immunize with MOG.
Yeah.
I will say, so one little detail that I left out is that these immunizations that they're doing are over and over and over and over and over. And this doesn't happen after like one immunization or one boost. They're doing like six boosts or 7. Something like that.
And they see the frequency of what they're describing increase. They backed it up to 3 immunizations, and they saw some, but not as many. Right.
So to me, it seems like you can immunize and boost, and those antibody affinities that are within your repertoire will go up very quickly. but that if you keep hitting over and over and over and over and over, you can birth these new specificities. Now that comes to one of their points where they say, what if there's a chronic infection or a non-healing infection and there's epitopes that are now blocked because you've generated some antibodies and they're not very helpful? You'd want to be able to generate different antibody specificities that could help you then fight that viral infection.
Right. Interesting.
So I think it still leaves a lot. Could we, does this contribute to autoimmunity? If it does, can we block it? Mm-hmm. Does it potentially lead to anti-tumor affinities? Could we increase that? Maybe checkpoint blockade is already doing that and we haven't looked for it. That's possible. Yeah.
Yeah. And so it's kind of neat. They have a framework that they present to us where there's a closed model and then there's the free diversity model so the closed model is what we learned in the textbooks, that it's somatic hypermutation that refines pre-existing specificities. And so this antibody scope is restricted to the VDJ repertoire that the antigen specificity is acting upon. But what they're saying is that somatic hypermutation can generate de novo affinities from non-specific starting points. And that the T cells can guide, they can provide signals, but even without initial BCR affinity, which is wild.
Yep.
Yeah. And okay, so the other implication that we talked about is that non-specific B cells can present antigen.
Yep. So
okay, sure. Okay.
So, right. Because we thought that B cells, the primary mechanism by which they take up antigen is receptor-mediated endocytosis by their BCR binding to antigen, pulling it in and digesting it.
So this is just what, like penocytosis, like just some stochastic grabbing of things around you?
I don't know.
Yeah. Yeah.
Yep.
Okay. I love this paper. I mean, it was just so fun to read. And and having this conversation, it really gets your mind working of like, it's bringing you back to old immunology lessons and then helping frame new frameworks. It's really cool.
Yeah. I think when I read it the first time, I went walking up and down the hallway and I was like, did you guys see this paper? I'm like, I feel like this is rewriting the textbook.
I feel like I'm going crazy.
True. What am I missing? Why is this not in like the top 3 journals? So I feel like I must be missing something.
Yeah.
So
that is always interesting, right? Because, for those, if you're in academia and you're in academic science, this is true, but like probably this maybe started up at Nature. But if your reviewer is a closed model, dogmatic person, it might be very difficult to convince them that this is true.
Absolutely. Absolutely.
And oftentimes because those people are topic experts, they get asked to review said papers. So you might be fighting that battle. It's hard to change the field. I mean, you really have to have a lot of evidence to change what we know to be true.
I think they used very, very clever models. I think they probably used the best available models. Mm-hmm. Is it a model? Are there caveats to it? Is there an issue with using a monoclonal BCR? Sure. Yeah. There's limitations. But I think the idea that it can happen is enough to warrant additional study into what's going on.
I agree. And I think the way that they could really further understand this in humans is people who are treated with CTLA-4, and then you pull out memory B cells with specificities that individual would have not experienced at the time of their CTLA-4 treatment, and see if you have to have a before and after, right?
That's the key. Yes.
You got to know if bystander B cells change mutation frequency after CTLA-4 treatment. Yeah.
I'm sure there's a way you could do large scale single cell sequencing before and after treatment.
And enough people are going on treatment, enough people are using these inhibitors that the cohort is probably not.
I think there's probably too much
noise, right? The polyclonal noise.
Yeah. So there's always going to be exposures to different things you eat, different things you breathe, different
right. You're not living in a bubble.
So
you're right.
I don't know. It will be interesting to see where the future of that research goes. Agreed. I like things that are like paradigm challenging and make you really think about, whoa, what did I just assume that I knew about immunology that might not really be true?
I love it. Love it.
All right. So that is Immune episode number 97. And I hope that some people have questions and comments because we know a lot, but there are limitations to what we know. And I thought the discussion was fun, but there's probably things we missed and we'd love to hear from you. Send your questions and comments to immune@microbe.tv. And again, if you like our chattering away or some of the interviews that we do or the other things we do at Microbe TV, please consider contributing to microbe.tv/contribute. Microbe TV is a 501c3. All of your donations are tax deductible and they go to keeping the lights on, to having our video editors help us, and to help Microbe TV keep bringing you reliable, interesting and exciting science. So, Stephanie Langle is at Case Western Reserve University. You're still Steph Langle on Blue Sky?
I am. I think so. I need to frequent that website more often. But yeah, it should still be Stephanie Langle at Blue Sky. Yep.
And I'm Cindy Leifer at Cornell University, and I am Cindy Leifer on Blue Sky as well. We're still on all those other things, but yeah, whatever. I am making a little presence. I call myself Immunominutes. So I'm starting to make some videos and do some things if you're interested.
Listen, if it's put, we're doing our best, right? You got to get in all the spaces. Yeah.
Yeah. Music on Immune is by Tatami. And thanks for listening to Immune, the podcast that's infectious. We'll be back next month.